All right, last video for the week. And for this video, we're only going to look at the first few, I think 14 slides of this. We're just going to look at the overview of matter and energy and then start talking about enzymes. So this particular topic goes with Unit 3, which was all about what we called bioenergetics and how living things utilize energy and chemical reactions to obtain energy. And there's a list of some things that you could see on the AP exam as far as processes. So cellular respiration, photosynthesis, fermentation, glycolysis, enzymes, and then gradients. So thinking about the proton gradients in photosynthesis to drive ATP synthase. ATP is our main energy source in a cell. And taking this back to what we just wrapped up in ecology, so ecology was the big picture of how energy enters an ecosystem as light, is converted to food in the plant, and then the rest of the food chain feeds off of that food from the plant the whole way up through the food chain. What we looked at in Unit 3 was the molecular stuff. So exactly how does a plant take light energy and convert it to something usable in glucose? And then when that plant does cellular respiration, how does that glucose get broken down to make ATP for cellular processes? Or when somebody eats that plant, how do they break the glucose down to get the ATP, right? So that's kind of like going big picture to small picture. We've looked at this from both perspectives. Some questions that you might see on the AP exam are listed here. Maybe take a minute, pause the video. We will hit on most of these throughout this video and then Monday's video. But if you have questions or have anything you want to ask me, pause here and then let me know. As you know, for each of these videos, the beginning starts with the vocabulary from this unit. Most of this, again, we will hit in this video and the video on Monday. But again, if there's something on here that you've never seen before, which that shouldn't be, but if there is something that you're not remembering, at the end of these two videos today and on Monday, if you're still confused, just reach out or attend the Zoom next week, and I would be more than happy to help you with it. Now, some things that we'll be hitting more on Monday, so we can save this particular slide for Monday, is the difference between a chloroplast and a mitochondria. We'll leave this for Monday because what our focus is going to be today is enzymes. Okay, so how enzymes function. So enzymes, if you think back to the lab that we did with the liver and the hydrogen peroxide, and remember you were capturing bubbles in a graduated cylinder. That lab was trying to get you to understand the functioning of enzymes. So the structure of an enzyme is a three-dimensional protein, right? So made up of amino acids all folded together. We usually just kind of see them represented as blobs. But the blob has specific shapes associated with it, with its active site most specifically. The active site is where the substrate attaches. And enzymes are extremely specific. They only attach one specific type of substrate. So for example, the specific enzyme amylase only fits with a carbohydrate. The liver enzyme that you were working with only fits with hydrogen peroxide. Lipase only works with lipids. So every enzyme has its own three-dimensional shape that gives it its own specific active site and its own specific substrate. Okay, so the active site attaches with the substrate, the enzyme does its work, and then it produces a product. The enzyme is able to redo a job, so once it's used, it's not done. It can continue to work and continue to work on substrates. And the other thing that we also talked about with our enzyme lab was the fact that enzymes are very picky with their environment. They have specific pHs and specific temperatures where they work best. And any of those changes in the environment can change that three-dimensional structure of the enzyme. Thus, the enzyme would no longer function. So pause your video here, read this prompt, and then on the next slide, they give you, the next two slides, they give you what you should answer. But remember, on this year's exam, we're not supposed to graph anything, so I did include the graph already done for you. So pause here, read over the next 3 slides, and then we'll go over the answers a little bit more in depth. Now notice with part A, it says to graph the data, which we've done. I've already done that for you, but it also says, and calculate the rate of reaction. So we'll look at how to do that. And then if we look at our planning our response, when you're looking at the data and when you're looking at the graph, you want to in your head just think about, okay, what is the relationship? What's going on here? So we see that for the first 30 minutes, we see an increase in the amount of maltose. But then after 30 minutes, we see that the product production kind of levels off. So thinking about what enzymes do, enzymes perform reactions on substrates. And I kind of use the example of stapling papers. If you put a stack of papers in front of me and I staple those papers, I'm going to increase my production rate. But after a while, what am I going to run out of? I'm going to run out of papers to staple. That's exactly what's going on with our amylase enzyme. So as it's starting, it's making more and more of its product. It's making more and more maltose. But then after a while, it's going to run out of its substrate. So we see that it's leveling off. So keep that in mind as we go through A, B, C, and D. So looking at the other part of part A with the calculate. Rate. Anytime we're doing rate, we're taking final minus initial over final time minus initial time. And that is on your formula sheet for you. So they want us to look at 30 minutes and they want us to do that from 0 to 30. So I like that the data is actually spelled out for me here. So I'm going to use this data. So when we're doing rate, we take the final concentration, which was 10.4 from our data chart, minus the initial concentration, which was 0, over final time. In this case, they wanted you to start at 30 over or minus initial time. So really, all you're taking is 10.4 divided by 30, which is 3.47. Now, I was very sloppy on here because I didn't include my labels. So let's go back and look at what those labels were. So we were looking at the concentration. So if I was doing this for real, I would have millimoles over minutes. So our final answer should be 3.47 millimoles over minutes. So that's our rate of enzyme production from 30 minutes or from 0 to 30 minutes. I apologize. Okay, so that's how we do rate. So we've answered part A. Now part B, explain why a change in the reaction rate was observed after 30 minutes. Well, if you kind of planned your response back here, you already know what was going on after 30 minutes. What happened? It ran out of substrate, right? So we kind of answered part B already. Part C says draw and label another line on the graph to predict the results if the concentration of alpha amylase was doubled. So if I increase the amount of enzyme, what's going to happen to the rate of the reaction? We would assume that the enzyme would work faster because there's more of it, and it would probably hit its max a lot earlier, but we would assume that we would level off at the same amount. So your line could look something like this. Again, this is just a quick sketch because I'm on my laptop. But that's probably what they're looking for when they talk about what happens if we add more enzyme. If this was this year's exam, again, you wouldn't be asked to draw a line because you wouldn't be graphing anything, but you might be asked to put that into words. So you could say something like the reaction rate would be higher, but the enzyme would probably reach its maximum production at an earlier amount of time. It would probably reach the same amount, the same concentration of maltose at the end and level off. Okay, so kind of putting that into words. Part D, identify 2 environmental factors that can change the rate of enzyme-mediated reactions and discuss how each of those would affect the reaction. So when you're doing this, some hints, they want 2, only use 2. They want you to identify them, and they want you to discuss them. So you want to make sure you're hitting both of those points. Things like temperature. What happened when you boiled the enzyme in our lab or you cooled the enzyme down? That's what it wants you to talk about. Or if we're talking about changing the pH, if you added an acid to this enzyme, what would that do to the enzyme? If you added a base to the enzyme, what would that do to the enzyme? And we know that anytime the environment changes, whether that's temperature or pH, that could affect the enzyme rate, right? It's probably going to slow down or stop the rate of the enzyme, because when the shape changes, we see the rate of the enzyme pretty much stopping or slowing down. So if you have the PowerPoint in front of you, this was copied right from the College Board. So the graph, which we don't have to do, the calculation for part A, part B is here, part C is here, and then part D is this last slide. So take a couple minutes, look over that, and that's really all you have to do for today, was just watch this video and take a look at that example problem. On Monday, as I said, we're going to go into cellular respiration and photosynthesis and review all of those reactions fairly briefly. And then we'll be done with Unit 3. So have a great weekend.